Alright, so picking up where we left off, we're going to talk about the second component part of photosynthesis. We're going to talk a little bit more detail about what's going on during the during the light reaction, the light dependent reaction, and the light independent reaction, also known as the Calvin cycle. And then we'll talk about some other extra topics towards the end as well, this lecture. Alright, so first off, kind of introduce you to the light dependent reaction. I have a little video here that's going to illustrate that.
In plants, photosynthesis occurs in specialized organelles called chloroplasts. The internal membranes of chloroplasts are organized into sacs called thylakoids. Surrounding the thylakoid membrane system is a semi-liquid substance called stroma. Photosynthetic pigments are clustered together to form photosystems. When a photon of light strikes the reaction center of photosystem 2, it excites an electron. Two water molecules bind to an enzyme at the reaction center. This enzyme splits the water and uses the electrons from the water to replace the electrons removed from the reaction center. Oxygen is produced in this process. The primary electron acceptor for the light-energized electrons leaving photosystem 2 is plastoquinone. The reduced plastoquinone passes the excited electrons to a proton pump embedded in the membrane called the B6F complex. Arrival of the energetic electrons causes the B6F complex to pump protons from the stroma into the thylakoid space, thereby generating a proton gradient across the membrane. Because the thylakoid membrane is impermeable to protons, the protons in the stroma must pass through the channels provided by ATP synthase. As protons pass through, ADP is phosphorylated to ATP and released into the stroma. This process for making ATP is referred to as photophosphorylation. When photosystem 1 absorbs a photon of light, its reaction center passes high-energy electrons to ferredoxin. The enzyme NADP reductase then transfers the electrons to NADP to form NADPH. Electrons lost from photosystem 1 are replaced by electrons generated from photosystem 2. A small protein called plastocyanin, PC, then carries the electrons from the B6F complex to photosystem 1.
Alright, so as I previously mentioned, we talked about these clustering of pigment molecules embedded in the thylakoid membrane. We call this clustering of pigment molecules a photosystem. Now, the photosystem consists of a centrally located, specialized pigment molecule called a reaction center complex. Alright, so your pigment molecules like chlorophyll A, B, your carotenoids, among other plant pigments, are responsible for absorbing various wavelengths of light, capturing that energy through the excited electrons in their pigment molecules, and then transferring those electrons to other plant pigments, which are eventually donated to a primary electron acceptor within this reaction center. Alright, so here you see your two photosystems. Alright, so photosystem 2 and photosystem 1. Alright, they capture light independent of one another. Okay. Alright, so here you're being shown an image of a photosystem. Alright, so you have your light capturing pigment molecules like chlorophyll, which eventually, once your chlorophyll molecule is excited by a photon of light, that energy, those electrons get transferred to neighboring pigment molecules in this photosystem, and where they are eventually donated to a specialized pair of chlorophyll molecules. Alright, and then you have these chlorophyll molecules that are excited, alright, in this reaction center. Alright, so notice that all these pigment molecules are embedded in the membrane of your thylakoid. Alright, so this is showing you those specific protein complexes, alright, the photosystem, embedded in the membrane of the thylakoid. So it's a highly complex structure. Alright, so Photosystem 2 and Photosystem 1. Photosystem 2 is the first in terms of is responsible for capturing energy that's later transferred to Photosystem 1. Alright, but in terms of discovery, Photosystem 2, Photosystem 1. Alright, Photosystem 1 was discovered first. Alright. Alright, so the reaction center of Photosystem 2, that specialized reaction center, is referred to as P680. Alright. And then for Photosystem 1, its reaction center is P700. Well, the reason for this is because it's all about the particular wavelengths of light or energy that they're able to absorb the best. Alright, so Photosystem 2, 680 nanometers, and then Photosystem 1, 700 nanometers in terms of wavelengths of light. Alright, so when these particular reaction centers are excited, alright, P680, when it's excited, alright, we'll just star it. Alright, now this is excited. Alright, same thing for P700. It accepts electrons. Alright, it gets starred. Alright, P700. It's excited. Alright, so this gives you a nice overview of the light-dependent reaction. Alright, it allows you to see the big picture. Alright, so we're going to walk through what's going on in each of these steps. Alright, but ultimately, alright, the whole purpose of the light reaction is to do three things. One is to produce NADPH. Alright, two, to produce ATP, and three, to produce oxygen gas. Alright, so these are the three major products of your light reaction. Alright, we're going to talk about how each of these three things is generated. Alright, so notice here that you have a kind of a linear flow to your electrons. They go from one complex to another, where they're eventually donated to this protein here. Alright, so this is the linear flow or design to the light reaction. Alright, we'll talk about cyclical electron flow a little bit later. Alright, so here, looking at Photosystem 2. Photosystem 2 absorbs light energy. These electrons are passed from one pigment molecule to the next, 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 until they're transferred to chlorophyll molecule P680. All right. So when this P680 reaction center is excited, all right, it becomes P680. All right, we'll star it plus. All right, it's gained electrons. All right, so now this is excited. It's absorbed light energy. It's captured light energy. Well, there's a problem. Because now there's been a loss of electrons from your original pigment molecules. So they have to be replenished. Alright, so in this case, water is split through a process of photolysis into electrons, protons, and oxygen gas. Alright, so this oxygen gas is the oxygen gas that we all breathe. Alright, so this is how plants produce and release oxygen, is through the splitting of water. Alright, and the whole process is to replace the electrons that were lost by photosystem 2. Alright, so in your membrane of the thylakoid, you have a series of membrane proteins, as well as a cytochrome complex. Alright, so these electrons get transferred from your primary electron acceptor, alright, your excited pigment molecule, to a membrane protein. That membrane protein will then transfer them to cytochrome complex, which allows for the movement of protons from the stroma into the lumen of the thylakoid. Okay. Alright, so we build up this huge concentration of protons in the lumen of the thylakoid. Alright, those electrons are then transferred from the cytochrome complex to this membrane protein. Alright, so plastocyanin. And photosystem 1. Photosystem 1 works completely independent of photosystem 2. So photosystem 1 is absorbing light energy. Those electrons, when excited, get transferred to other pigment molecules, alright, which are eventually transferred to your reaction center, P700. P700 becomes excited. Alright, now it's energized. Alright, so now you've lost electrons. So these electrons have to be donated from photosystem 2 to photosystem 1. Alright, so here you have the transfer of electrons from photosystem 2 to photosystem 1 to replace those that are lost. Alright, so what happens to the energized electrons that are captured by this primary electron acceptor? Well, you have another membrane protein, ferredoxin, that these electrons are transferred to. Alright, so again, thylakoid membrane. And those electrons are eventually transferred to a protein, an enzyme, in the membrane of the thylakoid called NADP plus reductase. Alright, so this enzyme transfers these electrons and hydrogen atoms to NADP plus, forming NADPH out in the stroma. Alright, so NADPH is synonymous to NADH and FADH2 in cellular respiration. Its job is to function as an electron carrier molecule. In this case, it's going to shuttle electrons from our light-dependent reaction to our light-independent reaction, the Calvin cycle. Okay, so looking at Photosystem 2 in a little bit more detail. Looking at how ATP is generated. Alright, so we have our pigment molecules in our photosystem that captures light energy. This energy gets transferred to our reaction center. Alright, our reaction center becomes energized. Alright, when it accepts these electrons, the electrons get passed to a membrane protein called plastic quinone. Plastic quinone transfers these electrons to our cytochrome complex, which allows for the movement of protons from the stroma into the thylakoid lumen. Alright, so you build up a large concentration of protons in the thylakoid lumen. Alright, so the concentration here is high, and here is low, alright, for protons. Alright, so that means the thylakoid lumen becomes more acidic, the stroma becomes more basic. Alright, so now you have a high concentration of protons in the lumen, low concentration outside the stroma. So you have a concentration gradient, you have a form of potential energy. These protons can move through our ATP synthase enzyme, and in doing so, moving back into the stroma, it allows for the mechanical energy needed to generate from ADP and inorganic phosphate ATP. Alright, so ATP is generated by photosystem 2 out in the stroma. Alright, photosystem 1. Alright, so photosystem 2, alright, is going to transfer its electrons to the cytochrome complex. The electrons are transferred from the cytochrome complex to plasticinin, another membrane carrier molecule, protein carrier molecule, electron carrier molecule. These electrons are carried to photosystem 1. Alright, they're there to replenish the electrons lost by photosystem 1 because photosystem 1 has absorbed electrons, energy. Those electrons are excited in our reaction center. They get transferred to our carrier molecule ferredoxin. Ferredoxin transfers those electrons to our NADP plus reductase enzyme, and those electrons are transferred to NAD plus to form NADH. Alright, so you build up a large quantity of NADPH and ATP out in the stroma. Alright, so photosystem 2 is responsible for making ATP. Photosystem 1 responsible for making NADPH. Alright, so let's look at cyclical electron flow. Alright, so in this case, some organisms only have the proteins to make up photosystem 1. They don't have photosystem 2. Or you have a plant that can utilize and switch from using photosystem 2 and 1 to just using photosystem 1. Alright, so here photosystem 1 captures light energy. That energy is transferred to your P700 reaction center. Alright, it becomes excited. Those electrons are transferred to ferredoxin. Ferredoxin transfers them to your cytochrome complex. In this case, you pump protons from the stroma into the lumen of the thylakoid. And those electrons get transferred back to plastocyanin, which are used to replenish those lost by photosystem 1. Alright, so it's one big cyclical process. Alright, and the whole purpose of this is to generate ATP. Alright, so in this case, you're not producing NADPH. You're only generating ATP. You also don't release any oxygen. Alright, so why exactly might a plant want to utilize this particular pathway or process as opposed to our linear electron flow? Well, what if you have more than enough NADPH? Well, then it doesn't make any sense for the plant to utilize that energy it captures into making something that it has more than enough of. Alright, the cell is always in need of ATP, so why not use the light energy to make ATP? It's pretty efficient. Alright, so one thing you should be able to do is compare the synthesis of ATP in oxidative phosphorylation to a synthesis of ATP through. So, talking about the mitochondria, we're talking about oxidative phosphorylation. And a chloroplast, we're talking about photophosphorylation. Alright. Alright, so you should be able to compare these two processes. Alright. So here, looking at the mitochondria, you have your mitochondrial matrix, you have your intermembrane mitochondria, you have your intermembrane space. In this case, your electron transport chain is capturing electrons transported by your electron carrier molecules. Here, whenever those electrons are transferred from one protein complex to the next, you pump protons into the intermembrane space, where you have a higher concentration of protons in the intermembrane space and a lower concentration in your matrix. Now those protons diffuse through your ATP synthase enzyme to generate ATP in the matrix. Alright. Now looking at photophosphorylation, you have protons that are moving from your stroma into your thylakoid space through your thylakoid membrane, and that proton gradient of higher concentration of protons in the thylakoid space compared to your stroma, protons diffuse through ATP synthase back to the stroma, generating ATP. Alright, so there's a similar mechanism involved. Alright, so now we're going to talk about the light independent reaction. Alright, sometimes this is called the dark reaction, even though it isn't an appropriate term to use anymore, as well as the more appropriate term, the Calvin cycle.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RuBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only one carbon atom from each molecule of carbon dioxide.
Alright, so the Calvin cycle. It is a cyclical metabolic pathway. Alright, but they're the opposites of one another. Okay? So, when you're talking about the citric acid cycle, or the Krebs cycle, remember that in the Krebs cycle, alright, when we're taking glucose and breaking it down into CO2, alright, we were oxidizing it. Okay. In the Calvin cycle, alright, we're taking CO2 and generating, alright, G3P, which is a precursor for glucose. Alright, in this case we're going to be reducing CO2 into this sugar. In this case, we were transferring energy to make NADH and make ATP. Here, we're going to be doing the opposite. We're going to be using energy that we captured from the light reaction and ATP to reduce CO2 into G3P. Alright, so Krebs cycle is catabolic. The Calvin cycle is anabolic. Okay. Alright, so the Calvin cycle can be broken up into three main stages. Alright, so here we have first carbon fixation. Alright, so in this case you're taking a inorganic form of carbon dioxide, and fixing it, attaching it to a pre-existing precursor molecule, alright, which we call RUBP. Alright, ribulose bisphosphate, and generating a 6-carbon molecule. That 6-carbon molecule will then split into 2 3-carbon molecules. Alright, which we'll call PGA. Alright. PGA will then be reduced to glyceraldehyde 3-phosphate. Alright, so this is where your ATP and your NADPH comes in. Alright. And then the RUBP that we use at the beginning of the process has to be regenerated. Alright, so a majority of this glyceraldehyde 3-phosphate that we make is used to regenerate RUBP. Alright, so this is very similar to the Krebs cycle. Okay, so recall in the Krebs cycle we had our little acetyl group, alright, which was 2 carbons. Alright, it combined with our oxaloacetate, which is 4 carbons, to make a 6-carbon molecule of citrate. That citrate was oxidized and reconverted back into oxaloacetate, or in terms of your Krebs cycle. Here we're doing the opposite. Alright, so here you have your ribulose bisphosphate, or REBP. Alright, so this is 5 carbons. Alright, it has 2 phosphate groups on the end of it. It combines with CO2 to form a 6 carbon molecule, which splits into 2 3 carbon molecules. Alright. Now, notice here, they have 3 CO2 molecules entering, they have 3 REBP molecules entering, so you're forming 3 6 carbon intermediates that split into 6 3 carbon intermediates. Alright, so you can get 6 PGA molecules, or 3 phosphoglycerate molecules. Alright. But only 1 carbon dioxide molecule can enter the cycle at a time. Interact with 1 REBP molecule to generate the 1 6 carbon molecule that splits into 2 3 carbon PGA molecules. Alright, but for simplicity's sake, they've gone through and shown you what's going on with 3 CO2 molecules entering the cycle. Alright, so this is our carbon fixation stage. Alright, so next we have our reduction phase. Alright, where this 3 PGA molecule, you attach ATP, you attach phosphate groups from ATP. Alright, so here you attach phosphate group. So now it's bisphosphoglycerate, alright, because you have 2 phosphate groups instead of 1. You transfer electrons and hydrogen atoms from NADPH to 1,3-bisphosphoglycerate to generate glyceraldehyde 3-phosphate. Now, at the end of this process, you make, for every 3 CO2 molecules that enter, you make 6 G3P molecules. Alright, so 6 3-carbon G3P glyceraldehyde 3-phosphate molecules. Now, one of those molecules can leave. The other 5 are used to regenerate REBP. Alright, so in this case, it would take 6 CO2 molecules to make 1 molecule of glucose. Alright, which fits really nicely to our balanced chemical equation, alright, because it takes 6 CO2 to make 1 molecule of glucose, where this is where your 6 CO2 come in. Alright, so every 3 CO2 that enter the cycle, you get 1 G3P molecule. So it means it takes 2 G3Ps to make 1 molecule of glucose. Alright, so here our plants are capturing light energy from the sun and storing it as chemical energy in the form of sugars. Alright, now the sugars are mainly going to be sucrose. Glucose as well, but sucrose is the major sugar you find in plants. That sugar can either serve as a form of potential energy, chemical energy, or it can be used to make cellulose or starch. Alright, so we can have a storage or structural role. Alright, so a lot of this potential energy is stored in roots. All right, tubers, seeds, fruits. And then you also release oxygen gas into the atmosphere. All right, which ties in back with the carbon cycle. All right, so this is showing you kind of the big picture of photosynthesis. All right, so it's showing you how CO2 is entering the leaf. All right, CO2 that enters the leaf is a input for the Calvin cycle. The water the plant takes up through the roots gets transported to the leaves. All right, is going to be used in your light reaction. All right, through photolysis, your light reaction releases oxygen gas. So this occurs in photosystem 2. All right, this is the oxygen gas that leaves the leaves into the atmosphere. All right, the light reaction is responsible for synthesizing ATP and NADPH, which is used to reduce CO2 from the atmosphere into G3P, which is used to make sucrose or starch or glucose or cellulose. And then that sugar is exported from the leaves to other tissues of the plant. Now, normally, all right, your C3 plants, they'll carry out the Calvin cycle. All right, they'll take in CO2. The CO2 is combined with REBP to generate PGA. The enzyme involved in this fixing of CO2 to REBP is called ribisco. Now, ribisco will function in the absence and the presence of oxygen. Now, everything's okay when CO2 is widely available, right? But when the plant doesn't take up CO2 and oxygen is the predominant gas, all right, the oxygen combines with the REBP to form PGA, and this PGA is going to be reconverted back into REBP. But in this case, you're using the ATP and NADPH to release CO2. All right, you're not actually making any sugars. All right, so you would think that this would be a wasteful process. All right, that's what we call photorespiration. All right, so this is the Calvin cycle when everything is normal. All right, you take your CO2, you combine it with your REBP, ribisco converts it to PGA, you get your G3P, and you make your sugars. All right, here you're not making any sugars in photorespiration. All right, so your C4 plants. All right, so they try to minimize photorespiration. All right, so they take the CO2 and they convert it into a 4-carbon compound. All right, so when you're looking at the structure of C3 versus C4 plants, your C4 plants also have these bundle sheath cells that are tightly compacted around your veins of your leaf. All right, so your xylem and your phloem. And then your mesophyll cells are loosely compacted or between the bundle sheath cells and the leaf surface. All right, so to give you a little bit of a better picture of this. All right, so here you have your C3 plants, which make up a majority of the plants on this planet. All right, you have your mesophyll cells, all right, interacting with your bundle sheath cells and in your vasculature, but they're not as tightly compacted. All right, here in your C4 plants, your mesophyll cells, there's a ton of them that are adjacent to your bundle sheath cells. All right, they're more numerous and connected to these bundle sheath cells. All right, so keeping this in mind, the structure, the tissue structure of this plant. Alright, so in the mesophyll cells of your C4 plants, you have an enzyme called PEP carboxylase. Alright, so PEP takes up the CO2 that the leaf brings in through its stomata, and it has a much higher affinity for CO2 than Rambisco. Alright, so it can fix CO2 even when CO2 concentrations aren't very high. Alright, and this 4-carbon intermediate molecule is then shuttled into the bundle sheath cells. Alright, once in the bundle sheath cells, the CO2 is released and then used in the Calvin cycle. Alright, so here you're looking at your C4 plant tissue structure. Alright, so your mesophyll cell brings in CO2. This CO2 is fixed into a 4-carbon molecule. So you have PEP, phosphoenolpyruvate. It combines with CO2, right, through the enzyme phosphoenolpyruvate carboxylase to form oxalo acetate. That should look fairly familiar. Alright, oxalo acetate is converted to malate, which is shunted into your bundle sheath cell. You release CO2, you're left with pyruvate, which re-enters the mesophyll cell and phosphorylated into phosphoenolpyruvate. The CO2 in the bundle sheath cell enters the Calvin cycle. Alright, so here, comparing your C3 to your C4 plants. Your C3 plant carries out the light reaction and the Calvin cycle in the mesophyll cell. Alright, your C4 plant carries out the light reaction in the mesophyll cell and carries out the Calvin cycle in the bundle sheath cell. Alright, so there's a different location for where these two processes take place in your C4 plants. Now, your C4 plants are things like corn and sugarcane, for instance. Your CAM plants, also known as your crustacean acid metabolism plants, or CAM for short. Alright, so your CAM plants, they are typically found in very arid, dry environments. Alright, so they only open their stomata at night in order to minimize water loss. Alright, because in these particular environments, like a desert, at night, the temperatures get quite cool. Alright, so they don't lose as much water to the atmosphere through evaporation in this way. Alright, so they take in the CO2 at night and fix it into an organic molecule. Alright, during the daytime, the stomata remain closed, and then the CO2 is released from your organic molecule during the daytime and is used for the Calvin cycle. Alright, so here you have our CAM plants. Alright, so things like cacti or agave or pineapple or orchids. These are all CAM plants. Alright, so CO2 is fixed into a organic molecule at night, And then during the daytime, that CO2 is released from that organic molecule and used in the Calvin cycle to make G3P. Alright, so there's a temporal or time difference. Okay, so this is carried out in the mesophyll cell, right? But carbon fixation occurs at night. Carbon reduction occurs, as well as a regeneration of REBP occurs during the daytime. Alright, so this is a nice little comparison for C4 and CAM plants. Alright, so our C4 plants, alright, CO2 is fixed into an organic molecule in the mesophyll cell. It's transported into the bundle shoot cell, CO2 is released. It enters the Calvin cycle, you get sugar. Alright, so there's a spatial separation between where our light reaction and dark reaction take place, between where CO2 enters the cell and is then used for the Calvin cycle. Alright, so there's a spatial difference. There's a temporal separation. Alright, CO2 enters the cells of our CAM plants at night. It's fixed into an acid during the daytime. That acid is then releases CO2 during the daytime, which enters the Calvin cycle. Alright, so there's a temporal difference in the steps involved in the Calvin cycle. Alright, so you should be able to compare C3 to C4 to CAM plants in terms of where each process takes place and some of the advantages that your C4 and CAM plants have over C3 plants. Alright, so this is a nice big picture of looking at everything involved from cellular structure to cellular physiology to the assembly of proteins to diffusion, osmosis, and active transport, and the production of energy through cellular respiration or photosynthesis. Alright, so this ties everything together. Alright, so in terms of cellular metabolism, alright, this is it. This sums up everything. Okay, looking at the big picture. Alright, how everything is interconnected. Alright, so